morning, good afternoon, and good evening. Welcome to today's IQ webinar. I am Ling Zhang from Bristol Myers Squibb Company, and I will be your moderator. Today, we have 3 industry thought leaders to share the business cases on the current challenges and opportunities to establish dissolution similarity. Before we start, I would like to introduce our speakers. Andreas Abend. Andreas holds a PhD in organic chemistry from University of Karlsruhe in Germany. He is currently a director in Merck's Analytical Science Department, leading a group of scientists supporting API and drug product development. He is a member of Merck's Biopharmaceutical Advisory Team, PQRI's BTC, and a member of IQ's Analytical Leadership Group. Andreas is one of the leading authors for several publications on dissolution, clinical relevant dissolution specification. Okay. Doris Angela Dess is a director team leader in Pfizer's Global Regulatory Affairs. She holds a bachelor's degree in chemistry from Parleman University and an MBA from Rensselaer Polytechnical Institute, New York. She has 25 years of pharmaceutical industry experience in a variety of disciplines, including analytical research, clinical supplies, sourcing, and regulatory affairs. She is a member of Pfizer's Dissolution Advisory Council, a sub-team leader for the IQ Dissolution Working Group, and a past chair of AAPS Dissolution Community. Connie Langer is a director at Pfizer's Global Product Development in Groton, Connecticut. She holds a bachelor degree in chemical engineering and an MS degree in oceanography from the University of Connecticut. Connie is currently a regulatory strategist in the Global CMC Department, accountable for providing global strategy and the regulatory submissions across the lifecycle. She is a member of the ICPE PQLI ICHQ-12 working team and co-chairs the IQ ICHQ-12 working group. So without further ado, I would like to pass the floor to Andreas.
All right. Hello, everyone, and good morning, good afternoon, or good evening. And yeah, welcome to this webinar. Thank you so much, Limin, for the introduction. I will now go over the presentation outline real quick. Today, we'll talk about problem statements and business cases, why we need global harmonization for dissolution similarity assessment, and how is actually dissolution profile comparisons done, and how do we actually, approach this problem. Then we talk about challenges with dissolution profile similarity testing for IR products. You can easily translate these problems to modified release, extended release products, or even long-acting parenteral, etc. Any product for which dissolution testing and comparing results is important. Then we'll talk about dissolution profile evaluation and evolving opportunities based on a workshop, and I'll talk about this in a minute, where we look forward towards really progressing this field. Then we talk about opportunities and considerations for global harmonization, alignment on similarity criteria, acceptability of other approaches such as safe space, how we establish clinically relevant dissolution specifications, and how we can use the safe spaces then to establish acceptability of a product after certain changes were made in lieu of actually performing a statistical analysis or mathematical treatment of the data. And then Connie will talk about established conditions. So let's go into, oops, let me just advance the slides here real quick. Some acknowledgements first. First of all, I mean, this presentation would have not been possible without the help and the input from many, many people. First of all, we wanted to thank the organizers and the participants of the workshop that was held at the MCERC at the University of Maryland in Baltimore in 2019. This workshop was titled In Vitro Dissolution Similarity Assessment in Support of Drug Product Quality, What, How, and When. And out of this workshop, you will see several publications that address this issue. Publications on mathematical treatments of dissolution profiles, as well as suggestions for how we actually work towards a global harmonized guidance. Also wanted to thank the IQ Dissolution Working Group members. Over the past several years, we had very good discussions on several topics related to dissolution, including similarity assessments. And a special note, a special thanks goes out to Michael Cohen from Pfizer. He just retired. He's been one of our very trusted, very insightful members, and he has significantly contributed to many of our papers and presentations. And then also the reviewers of those manuscripts, as well as the presentation material that you see today. So with that, I want to start with the problem statement. So really, the requirements to demonstrate the dissolution profile similarity is really different across regulatory agencies in different parts of the world. And this is really a challenge because for an industry that is operating globally, demonstration of product similarity from one market to the other could be very challenging. It could actually be questionable whether or not you can make desired changes or needed changes in your manufacturing process and not knowing whether or not these will be accepted in all markets. Because we typically don't make different variants for different markets. This can cause unnecessary delays in new product approvals and approval of post-approval changes, which is really what we're talking about today mainly. And that's why this is really desirable to move forwards towards global harmonization. Of course, we're not doing this just from the perspective of the industry. We really think that if we accomplish a globally harmonized guidance document and set of rules, we benefit patients as well as the regulators and the industry. So for the patients, of course, it would really hopefully allow them to have access to drug products quicker and hopefully at lower cost. And for the regulators, the speed of new medicines to the markets and acknowledging that other regulatory agencies have already reviewed changes or approaches which rely on dissolution profile similarity, but not only on dissolution profile similarity, are acceptable from a quality perspective. And you can think about reducing that review burden significantly for regulators. And of course, for the industry, it's really efficiency and efficiency in applications, as well as making sure that we can efficiently move from one process, with acceptable changes, to another if deemed necessary. So with that, I want to actually go over very quickly why we're doing dissolution testing and why dissolution testing is so important in assessing drug product quality. And I really love this quote here from the FDA's 1997 Guidance for Industry for Dissolution Testing of Immediate Release Solid Oral Dosage Forms. And very quickly going through this one quote here about drug absorption from a solid dosage form after oral administration depends on the release of the drug substance from the drug product, dissolution or solubilization of the drug under physiological conditions, and the permeability across the gastrointestinal tract. Because of the critical nature of these first two steps, in vitro dissolution may be relevant to the prediction of in vivo performance. And when we talk about changes and when we apply dissolution profile similarity, it's mainly this third bullet here, where we want to make sure that in vitro dissolution testing can help us to ensure continuing product quality and performance after certain changes, and these are all listed in SUPAC, do not negatively impact product quality. And that is really, again, why dissolution testing is such a critical tool. Now, what is dissolution profile similarity really? Well, you can see I'm showing you here two profiles. One being a reference profile, and oftentimes we see for immediate release products that over time dissolution profiles shift. Most of the time we are concerned when this shift goes towards slower dissolution profiles, but that's not the only concern. You could also think about a shift in dissolution profiles going towards faster dissolution. So the concern here, first of all, with similarity testing is, well, does a product have natural variability? Or in other words, if I make a batch today and I make a batch another day, what's the difference actually in these dissolution profiles? And I could look at these profiles from multiple batches, and oftentimes they're very close to one another. It becomes more interesting to look at dissolution profiles when you make deliberate changes to your process. And these deliberate changes may be forced onto you. Let's say there's an excipient change, or you have to scale up your process, et cetera, et cetera. And then you want to assess whether or not that change still gives you a profile that's comparable to your reference. So the other issue here with dissolution similarity, of course, is, well, when are profiles not similar? And how do we decide to what point does similarity go, or where do we set a acceptance criteria? And we'll talk about this in a couple of minutes, just for one of the most popular approaches. But you can think, then further how this applies to more sophisticated approaches as well. So what are some of the challenges here? Well, challenges are typically when we do dissolution profile similarity in the time points and the variability of the dissolution data at these time points. These curves here do not show you any spread of the data, but per current guidance, dissolution profile or time points for dissolution profile comparison have to meet certain criteria, such as the percent coefficient of variation should only be or should be less than 20%. This is typically for the early time points. For other time points, the criteria is actually tighter. The other criteria that must be met is that the average difference between the amount released at different time points cannot be more than 15. That's another rule. And so you can see that these average differences usually at the beginning of our dissolution event are rather small. I mean, unless sometimes you can see bigger differences, but in many cases where drug products and the dissolution is erosion controlled, you're actually seeing smaller differences here. But in this point of your dissolution profile, the actual differences do usually increase. So we have a region here that's very informative about the rate of your dissolution profiles, but also an area where you can see probably larger differences. And then we have this end of our dissolution profile where the two profiles actually converge. And here, of course, the differences become smaller. So if you were to randomly pick time points, it would be nice if you could pick the time points here at the late end, because the differences here would be smaller. But again, that would not describe your dissolution characteristics of a product, and therefore you should actually consider using all of these time points. And that, again, in the current current regulatory framework, by applying our most favorite test, which is the F2 metric, that is challenging. So Other challenging situations with dissolution comparisons come from, for example, modified release products, where you perform testing first in an acid stage, now you're transferring this product into the actual media where the dissolution happens. Minute changes in film coating, for example, can lead to different profiles. And it also typically leads to high variability at these time points. One way to address a situation where the profiles are actually delayed a little bit is lag time adjustments. Most regulatory agencies do not even mention that option, with the exception of the PMDA. So again, this is something that can be challenging and makes assessment of dissolution profiles very difficult. I mean, think about the difference here between these two profiles. If you just look at these at the same time point, the difference here is about 50% label claim in this order. So one would probably lean towards saying these are not similar. However, if you lag time adjust, they will actually be very close to one another, and similarity can be thus demonstrated. Another challenging situation is when profiles cross. In this situation, you can argue that test and reference products have different in vitro dissolution properties or different steps in the dissolution. Something has changed here. However, if you were to blindly apply, for example, the F2 metric, it will tell you that the output of your F2 calculation, which I will show you in a couple of minutes, is going to be acceptable. However, I would be very careful with crossing profiles and trying to assess this by a method such as the F2 test. Okay, now we're talking about the F2 test. Most people that are attending this webinar are probably very familiar with this test, and it essentially centers around this equation here. And then this equation here, which can be described or termed quadratic mean difference squared, you are essentially looking at the squared difference between your reference and your test profiles at different time points. And the value that you're getting for F2 depends on how, or the value that you determine here on your quadratic mean difference. If that quadratic mean difference, for example, is 10, let's take that for an example, then the square of the quadratic mean difference that you plug in here is 100. And now you're dealing with the square root of 101. If we round this down a little bit, the square root of 100 is essentially 10. And now 100 divided by 10 is 10. So in other words, you're calculating the log of 10 to the base of 10, which is 1, and you're going to end up with an F2 value of 50. If your quadratic mean difference, if this distance difference here is less than 10, F2 will be greater than 50. And if your difference is greater than 10, your F2 will be less than 50. So this is failure, still acceptable, failing. And of course, if you get a F2 value of 100, it means this term here essentially is 0. And that would always, make you cautious whether or not your measurements were done appropriately or your method is discriminating at all. So if we can't apply F2 because of the reasons that I just explained earlier, and that has to do with the choice of time points, allowable time points for your dissolution curve, as well as the variability in your time points, we resort typically to model-dependent or model-independent statistical tools. In regulatory guidance, you find F2 bootstrap or bias-corrected F2 bootstrap, as well as the Malar-Nobis statistical distance, MSD, or the ACLMND, which is the approximated confidence limit for the Malar-Nobis distance, and some other tests, in-house tests that I'm not going over, but these may all be acceptable to regulatory agencies. At this workshop, we actually discussed in depth other more novel approaches. They are not novel, but they've been around for several years, and they have distinct advantages over these tests here. And you will see in our publications that are coming up, hopefully towards the middle of this year, what the characteristics of these tests are, as well as when you should apply those. So more on this to come. Now, if F2 fails, you're typically stuck with either rationalizing why failing doesn't matter, or you may end up running a BE study. And with this, I'm handing it over now to Doris.
Thank you, Andreas. With that, the next steps is to discuss the dissolution similarity assessment. Dissolution profile similarity evaluation typically is used for the drug product development, post-approval changes, to support requests for biowaivers, multi-source products. But how we determine the similarity evaluation? We just heard from Andreas that we have to demonstrate that we have a similarity in the curve and the selection of time points. But we have to ensure that the testing is conducted under identical conditions. We also have to ensure that we have 12 dosage units for both the reference and the test product. And when we are constructing the curve by plotting it, we have to use the mean percentage of the drug dissolved relative to the product level claim. The same for the test and reference products will be determined by calculating the similarity F2 factor that Andrea just discussed. It's very important to highlight that the dissolution profile F2 is required to support some post-approval changes, but we could consider a safe space in lieu of it, and in other slides we will explain how we can take advantage of once we establish safe space to use as acceptability test in lieu of similarity. Global dissolution similarity testing is very important when we are preparing the dissolution strategy, that we design all our testing in terms of the global regulatory markets that we are intended to use. And for that, it is imperative at the moment to review what the regulatory requirements for the countries that we intended to file are expecting in terms of the acceptability of the dissolution similarity package that we are providing. And some considerations that we need to have when we are designing this dissolution regulatory strategy is that we need to obtain information about the biopharmaceutical classification system, understand what will be considered appropriate selection of a sampling time points at the early time points, how we can construct and characterize the dissolution similarity curves, and also the selection of the time points. What will be the dissolution conditions, the medium, the agitation, if that will be acceptable to the regulatory agencies. Specifically, do we have the compendial method that is requested in that country, or we should file using the approved QC method, or if the safe space methodology is acceptable in that country. Typical F2 similarity criteria, as Andrea has mentioned, is at least 12 units should be used for each profile determination. A minimum of 3 points are required in some countries, and I will let you know later on that it varies, these minimum requirements by country. The time point should be the same for the two products. only one measurement should be considered after 85% dissolution for either of the formulation. The mean dissolution values can be used if the percent coefficient of variation is no more than 20% of the early points. I would like to highlight that that's important in the early selection of the time points because if we have a sample point at 5 minutes, sometimes we have variation up to 40%, and it's very important to have the early selection of the early time points. And then in that case, we can actually select the first point up to 10 minutes, and that's what is acceptable at the moment in the majority of the regulatory guidances. If we have to make sure that we don't pass 10% at other time points, like 20 minutes, 30 minutes, that the difference should not be more than 10% according to the country. When F2 is not suitable, alternative methods to establish similarities are considered acceptable when indicated in the guidance. I wanted to highlight that because at the moment, certain guidance are silent in what will be the acceptability of the methodologies. And in those cases, regulatory interactions is necessary to ensure that the proposed alternative method will be acceptable. In 2016, we start establishing a comparison of all the guidance documents that are listed here. And recently, we also evaluated the ICH N9 to ensure that we actually establishes the divergence and convergence on the similarity requirements, specifically the number of batches that we have to use, the media selections, what will be the criteria to establish the similarity requirements, and if all the countries accept that if we have values within the first 15 minutes above 85, if the F2 calculation is exempt, and specifically regarding the minimum number of time points to generate the F2 calculation, and how countries define the last time points for an F2 calculation. And to our surprise, this exercise actually led us to find significant difference in certain parameters, specifically regarding the minimum time points. In Brazil, it's required the minimum time point must be 5. But if we are designing our resolution strategy for the United States markets, the guidance state that minimum will be 3. So it's very important that when we are designing the global dissolution strategy, we take into consideration the acceptability of the minimum time points. You will see otherwise that countries like Mexico and Brazil require 5 points, and other countries like Canada actually want a demonstrations of what will be the minimum adequate time points. So there is no specified. So therefore, we need to ensure that we know upfront in which countries we are going to be filing, so we can design our resolution strategy with different disciplines and build our CNC package accordingly. I also wanted to highlight that in the United States, not all the post-approval changes require multimedia dissolutions, and it's very important to always check what the guidance states so we can build our package accordingly. Andreas highlighted some information about acceptance of alternate statistical models, what models are acceptable, and there are a variety of models, dependent and independent. And I wanted to highlight this, that when we were gathering the data from different regulatory agencies, it was noticed that in some countries, only the model independent, that means the F2, is the only acceptable way to compare dissolution profile. In other countries, the expectation of the agency is to have the bootstrap F2 and the Mahalanobis distance. And in some cases, recently in the workshop in 2019, we learned that in Canada specifically, more than one model might be requested at times. But I wanted to highlight here that although Brazil is indicated F2 has been the only only method that can be accepted. We have example within the industry that alternative statistical methods were acceptable in Brazil once the in vivo data was performed, and that required regulatory interactions and multiple discussions with the agency in order to be accepted. So it's very important that we don't assume that we can use any of the statistical methodology. It's imperative for us to check what the guidance requires in order for us to be in compliance and avoid some rejection. It's also important to highlight that evidence of the statistical software that has to be validated and in compliance might be required in the EU and Australia, between other things. Another difference is that we notice, and that you can see here, there is a wide divergence in the requirement of the last point. How is determined? In United States, for instance, both the reference and the test product have to reach 85%. In EU, any of the reference of the test product after reach 85% can be selected. And you can see that it changed from country to country, and there is divergence in which how to determine the last bullet point. And this also might change how we generate the report, and you can easily see that we will have to customize our dissolution package and have some assurance each time that we are going to do the filing in those countries, just to ensure that we are in compliance with the requirement, because it might be cases that the values might be different. Something that is a very good news is that we are now with ICH M9, no more than one mean value of more than 85% for either of the products. So one strategy is to use the ICH M9 to define what is acceptable in those countries, and also verify if those countries are signatory country of ICH. So with this, I wanted to highlight what are the convergence and divergence on similarity regulatory requirements. The convergence is the number of doses units for both reference and test products is the same. The F2 criteria, they have to be above 50 to between 50 to 100 is acceptable for all the market that we assess. The criteria for when F2 mathematical evaluation of profile similarity assessment is unnecessary after reaching more than 85% dissolved is similar in all these countries. The divergence specifically is the time point selection, which I highlighted before, the selection of the early time points and the last time point that differs in many countries. The percentage of coefficient of variation also changed. In one country might ask less than 20% for the second, third, and fourth points. There are countries that require the coefficient of variation to be 15% for all of the points. And that means that it will pass in one country, but it will fail in another. And as I mentioned, the acceptability of alternate statistical approaches is not acceptable in all the markets. So where are we now after the workshop that we have in 2019? And what are the discussions that we are trying to further and advocate specifically to get this dissolution similarity criteria as a ICH topic? The F2 calculation has its pros. It's a mathematical calculation that is widely acceptable by all the regulatory agencies, but there are some differences. The test is very simple, it's easy to administer, and may serve as expedient first pass evaluations. The cons, that the F2 test cannot be used when the variability exceeds the recommended threshold. The F2 test does not provide information on the type 1 or type 2 errors. On the level of confidence that the profiles are or are not similar might change here. And since this is a secondary method, it's needed when the F2 test cannot be applied. So we start with F2, and we might have to use an alternative methodology because we have high variability. And the F2 test is a comparison of one profile versus another, and it's not an appropriate test for evaluating multiple batches of the test product versus the reference product, specifically during the early development where we have multiple batches and we have to do the comparison. So with that, we are proposing the implementation of the clinical relevance as a test that will be replaced in lieu of similarity. And that test will be considered as acceptability test. I will have now Andreas to do the presentation from here. Andreas?
Yep, I'm ready. All right. So Doris talked a lot about, the differences in regulatory expectations when applying F2. Of course, what we are not trying to do here is advocate for products or product changes that result in product with high variability. So that's, I just want to make that clear. But the question becomes now, well, if all our attempts of demonstrating that dissolution profiles are similar have actually resulted in non-similarity. And what are we going to do then? And that's where the implementation of clinical relevance of your specification becomes really important. The underlying assumption when we do dissolution profile similarity testing is, of course, that your dissolution profile is meaningful. It's kind of related to how a drug product dissolves in vivo, as I've shown you with this excerpt from the FDA's dissolution guidance. But if you don't have that link, your dissolution test is always going to be ambiguous. And also your acceptance criteria of your F2 equals 50 or greater. Again, this goes back to the difference in your dissolution time points. Is it on average 10? Then you will likely pass. If it isn't, well, then you're going to fail. So what are you going to do in those instances? And even all these advanced statistical or not so advanced statistical tools, they all use a equivalence margin or an acceptance criteria that is somewhat arbitrary. So we don't know where it's coming from. We don't know how you can justify this. It is been accepted and we all live with it, but we don't really know what its in vivo relevance is. So let's talk a little bit about clinically relevant dissolution specifications and how we can actually establish those. First off, it's really important to level set on definitions. So what are clinically relevant dissolution specifications? This is terminology that came out of a workshop at the MSOC that was held in 2017. And during this workshop, we defined that clinically relevant dissolution specifications are method conditions and the acceptance criteria that are based on clinical performance. So whether this is PK or more ideally PK and PD, you build a link between this in vitro test and in vivo performance. And with that, you can set meaningful specifications that allow you to reliably accept batches that are bioequivalent with pivotal clinical batches. So you're ensuring via this test that you have an adequate specification that allows you, or that will only allow product that has the same quality as that product that you developed is released to the patient. Now, in 2017, the IQ Dissolution Working Group came up with this clinically relevant dissolution specifications roadmap. And in here, we kind of suggest that sponsors really have to make this decision whether or not they invest in exploring the impact of changes of their formulation on in vivo performance via an IVIVC type approach or via modeling and simulation, physiologically based modeling, physiologically based biopharmaceutics modeling And in either case, these attempts are all geared towards establishing clinical relevance of the specifications as well as a dissolution safe space or safe spaces for other parameters, other critical quality attributes that you can measure and link to in vivo performance. However, you may not have to invest in IVIVC studies. You may have a process that is very robust, or you may have drug products containing highly soluble drugs like BCS class 1 and 3, and we saw ICHM9, for example, as a good example, where you can actually assume that clinical relevance is actually built in your spec without building this link to in vivo performance. If you do so, you will set your specifications essentially on precedented conditions. What that means is these are the conditions that you used to manufacture batches that are used or were used in pivotal clinical trials. We call this the traditional approach because probably more than 95% of products currently on the market actually followed this traditional approach, and our dissolution specifications in most cases do not have clinical relevance. And again, here we're following the current guidance when we then apply for our market applications and when we present what that dissolution specification should be. But this approach here is all about building that link between your in vitro performance, changes that you made, and then what is the impact on in vivo performance, either typically via PK or PK/PD. With that, I'm going to move on to another definition. We just talked about clinically relevant dissolution specifications, a clinical safe space or bioequivalent space. Again, these are the boundaries that are defined by in vitro specifications within which a drug product or all product batches are anticipated to be bioequivalent to one another. This comes straight out of the IVIVC guidance, or less optimally, but still possible, bioequivalent to pivotal clinical batches. Again, from a practical perspective, and I will show this in a couple of minutes, bioequivalent to your reference batch is really the ultimate goal, at least in my opinion. Now, is profile similarity testing always necessary? So do we have to do the F2 test? Again, you will see in the next slide actually where this is explained a little bit better, but let's say your pivotal batches were all showing this dissolution profile here. And you did actually study the impact of a formulation change on dissolution profiles, and you did also study this in vivo, and you found that this batch here actually shows you bioequivalence between this and this batch. So you can now assume any batch, any change that I'm making resulting in a dissolution profile that's within this space here is going to be acceptable from a biopharmaceutics perspective. So again, we're still meeting our specification depending on where that specification was set, probably set based on this curve, and we'll talk about this as well, as opposed to using for example, a dissolution specification set here at 15 minutes, because you know the product that will have a slower profile, and maybe with a dissolution specification set here based on the BE batch that was used, or your variant batch, whatever you want to call it, it's still acceptable. So, but just keep in mind that this entire space here is your dissolution safe space. And depending on what parameter you've used in making this batch, you can now also use the materials attributes that you can determine based on this batch to show that these levels, or whatever that acceptance criteria was, is truly acceptable from an in vivo perspective. So it doesn't only apply to dissolution, it applies to other specifications, other materials attributes like the particle size distribution, for example, as well. If you followed your dissolution specification setting based on approach 1, just using your pivotal clinical batches, Usually these clinical batches all have very similar dissolution profiles. And you're going to end up with setting a specification essentially, based on guidance. Here I picked a dissolution time point where we have more than 85% release, following EMA's position paper from 2017, subtracting 10% gives me a Q of 80 in 30 minutes. Now, with this information, I can also just simply calculate, well, what is my F2 space? What if I'm making changes relative to this bio batch, or one of these clinical batches typically is denoted as your bio batch? If that is the case, well, that change should not be more than 10% on average in these dissolution time points. Again, typically, a change will result in a slower or a faster curve. That's why 10% from these here is really what I'm looking for. But this would be your F2 space. Of course, as Doris has explained, you cannot use these later time points, so you're probably stuck somewhere here with the selection of the time points that you can use. Now, had you invested in safe spaces, and you have shown that, for example, batches that have a slower profile are still bioequivalent to this batch here, which is your reference batch, your bio batch, then you can use this entire space here to justify changes, and you can use that to demonstrate, or you've already pre-demonstrated that these batches would be acceptable. So this safe space gives you a lot more flexibility in a post-approval world. All right, so considerations of using the safe space in lieu of F2. Again, to me, the other thing that I didn't mention here, but I alluded to that at the beginning, is this acceptance criteria of 10, or an average of 10, is somewhat arbitrary, right? If you have established a clinically relevant or a safe space, now you actually know what that difference really should be. So you can set these equivalence margins appropriately. And I just leave it there because there's more to just comparing profiles when we talk about safe space. And we're still working on how we frame that. But again, setting meaningful acceptance criteria, if you had to do a statistical analysis of your data, is going to be a key element that comes out of your IVIVC studies, whether these are via PBBM modeling and simulation or traditional IVIVC approaches. Now, we don't know which regulatory agencies ultimately will accept safe spaces. The FDA does. Actually, they have shown at this workshop a decision tree of how they approach post-approval changes and whether or not these are actually flagging or would be acceptable or not, and whether or not the dissolution profile really flags that or not. But again, the take-home message really is that had you invested in a safe space and you know what your specification can actually detect or not, you should be using this, and it should give you more flexibility in the post-approval world. Available tools like established conditions is something that Connie now will talk about, but again, it's our position that we should be investing in clinically relevant dissolution specifications a lot more. With that, I hand it over to Connie.
Thank you, Andreas, and thank you, Doris, too. I think both of you provided a very clear picture of some of the challenges that we're facing as an industry when it comes to applying dissolution similarity testing in a global marketplace. I think that safe space approach that Andreas just described is really interesting, and I'd like to explore the possibility of leveraging this in conjunction with some of the new tools that have just come out and been made available through ICHQ 12. So in the next 5 slides that I'm going to share with you, I'm going to show you what that might look like in a real example. So before we begin, we need to build the foundation. So if we want to leverage a safe space, first we're going to have to ensure that it's supported by appropriate data set, like Andreas was just describing. So that includes method conditions and acceptance criteria that are based on clinical performance, where the goal is to reliably accept batches which are bioequivalent with the pivotal clinical batches. So we must also gain agreement from the Board of Health that the boundaries that we set in vivo specifications are acceptable. As he just mentioned, the United States is accepting this. Other markets, I think we're going to have to explore as we go. So the data to describe this safe space would be included in the 3 pillars that are on the left. So in P22, the dissolution method, as well as in P51 and P52 for the spec and method. And then once we have the safe space agreed, we could leverage this data to possibly reduce the regulatory reporting for potential changes that typically require in vitro dissolution testing and or bioequivalence testing. In the US, we refer to these changes as SUPAC changes or scale-up and post-approval changes as named in the guidance for industry. And these changes might include things like changes to components and composition that are described in P1, changes to sites in P3.1, and maybe changes to batch size that are described in P3.2. So if you look at the SUPAC guideline, these changes often require multimedia, multi-point dissolution, and possibly a BE study, as well as supportive batch data and stability data. So in addition to the pillars on the left, we add the 3 pillars on the right, the impacted section depending on what the change is, as well as the batch data in P54 and the stability data in P8. Then with the approval of the safe space, we would propose reduced documentation and or reporting categories through the use of a product lifecycle change management document, that's a PLCM, that's described in ICH-Q12, or possibly in a PACMP, which is a post-approval change management protocol for a specific change that you're planning. So for those not familiar with ICH-Q12, the PLCM document is a collection of established conditions and reporting categories that's reviewed and approved by the Board of Health. And once it's approved, it serves as a life cycle management guide that is applied to each unique product. Oops, I think we went too far. Thank you, Doris. So in this slide, we're going to show a few examples of composition, batch size, and site changes for immediate release products. Each row is split into assessment according to the current US guidance for industry for SUPAC type changes, and then our proposal if a safe space is in place. So if you're familiar with SUPAC IR guidance for industry, then you'll know that there are different levels of changes described in each category, and the documentation required in reporting categories are related to the risk to quality of the product. So we selected just a few changes for the purpose of comparison and illustrating our differences. So if we look at the first row, it's a change that is for excipient grade with no impact to stability or degradation. According to the guidance for industry, this would require multi-point dissolution profiles with conditions dependent on the BCS class. And it would require a prior approval supplement, which is the highest level of review for variation. Now, in contrast, if a safe space is in place, we would propose that if the established conditions are met, so the dissolution results for that post-approval change product fall within the safe space that Andreas just described, then this change could be made using an annual report and providing supportive batch data for the pre and post change product. So eliminating the need for those multi-point and multi-point dissolution profiles and reducing the reporting category significantly. I'm going to go to the next slide and show you how the flow diagram or decision tree that we use to guide our assessment and come up with some of these proposals. So in this, if you're looking at the bottom orange bubbles, the reporting categories there are shown in alignment with the Q12 guideline. So NL is notification low, and that's equivalent to an annual report in the US. And then, or it could be also a CBE 0. Then NM is notification moderate, which is a CBE 30, and then PA is prior approval, which is equivalent to a prior approval supplement. So just to orient everyone. So if we were to propose a change to something for composition, scale, or site, then the first question that we would ask in our decision tree is whether a safe space has been established. If the submission is for a product without a safe space in place, then you would just follow the guidance that pertains to the market where you want to file the change. So you'd follow the diagram down to the right. And then if a safe space is in place, you would follow the flow diagram to the left and perform a risk assessment to determine if a change in control strategy would be required. You might imagine that a change in composition could result in a change to the degradation products and stability of the product. In this case, you'd follow the flow diagram all the way down to the bottom left, and then you would include the data in the submission. So the reporting category would depend on what that change to control strategy was. So for example, if you had a new degradant that you needed to specify in your release spec, well, that would be a prior approval supplement or the highest reporting category. In addition to the supporting data around this new degradation product and justification of spec, the submission would include the pre and post change batch data for the product, but not necessarily the multimedia or multi-point dissolution. So then let's talk about the middle. If you would arrive here if you had a safe space and no change to control strategy was required. In this case, you could include batch data to show that you met the dissolution criteria defined by the safe space and include supporting degradation and stability data. If there were no expected stability and degradation risks, it could be made by annual report. If on the other hand, there was the risk assessment revealed a potential stability risk, then you might submit it using a CBE 30 or notification moderate. So we prepared a couple of detailed hypothetical scenarios to illustrate how this might work in a real life example, and I'll share those on the next slide. So the first example was for a change in technical grade of an excipient with no impact to stability or degradation. So if we start on the left with the change description, we could be a change from Avicel pH 102 to Avicel pH 200. This is actually one of the examples that provided in the FDA guidance for industry. And so for our scenario, we'll assume that the product is BCS class 2 and it does not have a narrow therapeutic index. In the absence of a safe space, this would be a prior approval supplement that would require multimedia and multi-point dissolution. So for our scenario, though, let's assume that the safe space was established based on a link between the in vitro release of the formulation variant and the in vivo exposure. Let's also assume that the risk assessment showed that the dissolution criteria of the post-change samples were met. And in addition, the product exhibited acceptable stability and there's no observed degradation. So based on this, you could conclude that the new grade of Avicel did not impact the performance or quality of the product. And then the change could be reported as an annual report, and the variation would include batch data to show that the dissolution acceptance criteria met, along with the supporting data that show there's no impact to the manufacturing robustness, degradation, or stability. So it would be a reducing both reporting category and evidence for this type of change. So in the second scenario that we prepared, it's similar. This would be a change from one site to another site in a separate location, but we would be using the same equipment, governed by the same SOPs and PQS. The environmental conditions and controls will be used in the process at the new site, and there's no changes to the batch records except for administrative changes, like address and so forth. So in the absence of a safe space, this would be reported as a CBE 0, along with an annual report for stability data, and the variation should include multi-point dissolution profiles using the approved method. For our scenario, let's assume that safe space was established and the risk assessment showed that the dissolution criteria for pre and post changes, the samples were met and the product exhibited acceptable stability at 3 months. In this case, the change would be reported as CBE 0 and include batch data to show the dissolution acceptance criteria was met. So in this case, the reporting category we're proposing is actually the same. And the reason for that is we would want to notify the agency right away if we're changing the site of manufacture. So it'd be notification low, but it would be immediate notification. But when the safe space in place, then the advantage is that you would just need to show the batch data that showed it met the safe space and not the multi-point dissolution data. So then we go to the next slide. So now it's my pleasure to summarize the work that the IQ Dissolution Working Group, and especially Andreas and Doris, have been doing over the past several years. So in our current state, as they described, similarity F2 criteria are widely accepted by global regulators, and they're used to support post-approval changes for products. However, as Doris was clearly mentioning, we as an industry are facing challenges with the global divergence of dissolution similarity criteria. By markets requiring different media time points and maybe different alternate statistical tools, it's very challenging to meet all the requirements with a single study. And you might imagine a scenario actually where different study design and criteria could result in a different overall conclusion, whether pass or fail. So this is just one of the reasons that the MCIRSE workshop was held in May of 2019. At that workshop, there was 160 scientists from academia, pharmaceutical industry, and several regulatory agencies. And it featured both podium presentations and interactive breakout sessions. The output from this session included an umbrella manuscript that was published in May of 2020. I think both Andreas and Doris referred to this, and if you haven't had a chance to read it, I would highly encourage you to do so. In addition, there are 3 more papers that are being authored to summarize the detailed discussions that happened during this workshop, and those are anticipated to be published later this year. And today, we have this webinar, and this is to bring awareness to the broad IQ audience of the challenges and opportunities that we face. The ultimate goal with all of these efforts are global acceptance of alternate approaches, including safe space, revision of regional regulatory guidance documents, and ultimately harmonization through acceptance as an ICH topic. And then if we, yep, thank you. The MCIRSE workshop really provided an opportunity for direct dialogue between regulators such as Anvisa, Health Canada, EMA, FDA, along with industry and academic stakeholders to identify gaps in knowledge and potential solutions. The papers that result from this workshop communicate broadly among the scientific and regulatory community potential strategies to overcome the challenges that we're facing. Meanwhile, while we're continuing to drive the science toward identification of the most suitable statistical and science and risk-based approaches, one example that Andreas advocated today was for the clinically relevant dissolution specifications and safe space. So advocacy is what we're doing today. We're raising awareness, we're sharing information and ideas, and having dialogue among key stakeholders. All of this is toward the ultimate goal of global harmonization and acceptance as an ICH topic. And then in conclusion, well, we know that we have divergence, and we're trying to make everyone aware with the goal of harmonizing the similarity criteria, identifying acceptable alternate statistical tools, and driving toward a science and risk-based approach. In the meantime, we're still living in the world where F2 remains the standard tool for assessing similarity. And we need to be really careful when we're designing and executing the dissolution studies that underpin the decisions on product modifications in this global landscape. As the science moves forward, we should look for opportunities to leverage the data to justify reduced documentation requirements and regulatory reporting categories with the goal of ultimately facilitating rapid patient access to safe, high-quality medicine. And ultimately, harmonization will create a common understanding for the dissolution similarity criteria and acceptable standards for statistical and science and risk-based approaches. So with that, I would like to thank the audience for your attention and time, and I would hand over the virtual podium to Lee Min, who will lead the panel discussion.
Okay, great. Great presentation. Thank you, Connie, Andreas, and Doris. So the divergence in dissolution similarity requirement is certainly an important topic around which getting alignment among the global health authority is historically difficult. So when all the stars aligned and F2 works well, life is good. All other situations, life could be challenging. So I would like to open up the floor for audience to ask a question. So you have options of typing your question in the question box. I see that we already have a lot of questions coming in. All right. So I am going to dive right into the first question. So have you been successfully in submitting a lag time justification in case of encapsulated comparator tablet formulation in case where F2 is less than 50 outside of Japan? Andreas, Doris, do you have any experience with that?
I have to say I don't. I have to admit this was one of the concerns that were expressed by one of our colleagues working in the generic industry. And yeah, so short answer is no, unfortunately, I don't.
Okay. All right. Doris, I know that you have been working in the regulatory industry, and
yes, the answer. No, I haven't had that opportunity outside of Japan and other markets. No.
Okay. All right. So perhaps we can, look into maybe consult with some of the internal, expertise, and if possible, we can always come up with a question. We can always come up with some response, and we can post the response in the IQ SharePoint site. So the second question, yeah. So the second question is, if the dissolution profile does not show 100% release, how do you interpret about the method? Do you think the method, is feasible to support the release, stability, specification, or?
Well, usually, I mean, a quality control method is typically developed to demonstrate complete release of your dosage form. And of course, the average amount dissolved is going to depend on the assay of your batch. So if you made a batch that was 95%, then you won't achieve full release. On the other hand, typically you should, though, your dissolution method should show you complete release of the dosage form.
Right. Yeah, so that is also one of the objectives when we develop a QC dissolution method, that we certainly want the method to be able to demonstrate complete release, and also have some sort of discriminating capability so that you are able to differentiate the batches that with different critical process parameter or material input. All right. What validation parameters are you performing for the validation of your method for comparative testing in 3 surfactant-free media, given you have the method fully validated for your QC media? Perhaps, Andreas, you have some experience. Yeah.
Well, again, I think what we typically would do is make sure that the detection method is adequately validated. So in other words, if you're using UV or HPLC, that method has to be demonstrating linearity, accuracy and precision. The actual dissolution method, I mean, that is, the condition is what it is. I mean, if it's multimedia dissolution testing at 50 RPM, that's what you're going to be working with. Of course, for a QC method, you look at other parameters. You're looking at the dissolution method itself, and you're trying to ensure that your variability at all time points is acceptable and that you are not measuring artifacts or any nuisance factors that you're not really interested in, because you want to have a method that is sensitive towards the solution and how the drug is released in vitro, but you're not really interested in some of the other aspects that could be contributing to your measurement variability, but that have nothing to do with the solution rate.
Mm-hmm. Yeah, I agree. All right, so for slide 21, for EU, there is a question and answer from the PK working group that recommend the bootstrap F2 over MSD. So could you shed some light on, the regulatory expectation in terms of using the alternative statistical tools?
So there are a couple of, if you really want to go into the statistics, there are other experts who can really answer this question. And one of them being the lead author of this day one paper from the workshop. So Thomas Affelder from Boehringer Ingelheim can answer this question very elegantly, especially since he has presented at workshops where statisticians from EMA were also present. I think the biggest concern that EMA has is that if you're applying the Molar-Hanovus distance, actually, if the variability in your time points is large, you're going to see the Molar-Hanovus distance shrinking, and you would make an assessment in favor of dissolution profile similarity. That is not, again, that is questionable. And that's why the F2 bootstrapping approach does not have this liability, but it has other liabilities. So that's why we also advocate for really using more novel, appropriate approaches to demonstrate whether or not things are similar or equivalent or not. I hope that answers the question. If not, please send me an email and I will get you the references that you're looking for.
Great, thank you, Andreas. Yeah, so another question is about the lag time. If the lag time adjustment is not mentioned by other authorities and it is only accepted by PMDA, does that mean it is only accepted by PMDA? Do you have any experience varying the adjustment of lag time with other health authority other than PMDA?
I'm not sure if we had any, maybe our regulatory affairs CMC group could weigh in, or Doris, Connie, you may have some experience, but we have not done lag time adjustment because, not that I know of, but certainly it is something to look into, especially when you see profiles that look very similar and it's just the rupture either of a capsule shell or film coating that gives you slower profiles, but the overall release, let's say of a granule inside of a capsule, may still be the same. So there are ways to address this issue other than with lag time adjustment, but I think lag time adjustment is a very attractive way of dealing with this problem.
Yeah, I would just add on to that. I guess I'm pretty, based on the guidelines, Korea would also accept this approach. I don't have any direct experience with filing that in Korea, but I think that they are also open to the lag time adjustment.
Yeah, yes, Connie, I wanted to say that the lag time actually in PMDA guidance as well as Korean guidance, they specifically indicated how to do the corrections and everything. In other regulatory agencies, I have not seen the description, or there is no experience regarding the acceptability of the lag time corrections. So I think the question was, do we have experience in other markets other than the PMDA, only the Korea? Because if you take a look at the regulatory guidance, it's similar to the Japanese guidance.
Okay, thank you. The next question is really about using in silico model to demonstrate the similarity of the drug product. So do you have any experience with the submission of in silico bioequivalency, you know, referring to FDA guidance in 2020 on use of PBPK models for drug product quality assessment? So if your in vitro dissolution comparison shows F2 less than 50, which is demonstrate in similar, but if you are able to establish your PBPK model and demonstrate your AUC and Cmax fulfill the bioequivalence criteria, have you seen the success on that approach?
So there were a couple of workshops. Again, in 2019, there was a workshop that was all about modeling and simulations. And at that workshop, presenters had actually shared their success stories with modeling and simulation approaches. That is still evolving, right? There's new criteria coming in. How do you actually validate the model? And it's coming back to, well, you got to validate this based on some sort of an IVIVC study as well, which is, of course, now shifting a little bit to this more traditional IVIVC landscape, but we'll see how this evolves. I think it is an exciting field. What we will learn is going to be based on more or less experience, because no drug product development is the same. So you will always see differences, maybe differences in parameters that impact your in silico model. I think, again, whether or not I can give you a solution that in this case it works, in the other case it doesn't, it's going to be more or less based on the clinical data that you have to establish these models.
Yes, I would like to comment that something that I learned through all these workshops with the regulatory agencies is that they definitely want to get involved in the assumptions when they are establishing the models and making sure that there is alignment and agreement upfront prior to actually presenting the data. So I think it's very important to highlight that having early regulatory interaction is critical with these novel approaches. And I believe that now we have a variety of assumptions that can be made, but if we have those alignment upfront, that will help with the regulatory filing and specifically advancing the acceptability of these models.
Yeah, great. Okay, so how widely that safe space are accepted, you know, during the global filing? You know, I have a similar question, you know, do you know how many countries that are currently accept a clinical relevance spec or safe space approach?
I mean,
any ideas?
again, I don't think there are large enough data sets available that will tell you in what cases specific countries have accepted the safe space arguments and when they haven't and why they haven't. But again, the FDA is very open to this concept. So Health Canada, at least the representative from Health Canada at our workshop, said they will take it into consideration. EMA definitely will take this into consideration as well. Again, there are nuances that I was trying to explain to you earlier with IVIVC, level A IVIVC. Does all of that need to be met? And, you know, I could see that there's still disparity even in what your clinically relevant dissolution, what that specification, the method conditions should be. So there's still a lot to learn here, but directionally, it seems that many more modern regulatory agencies are all, willing at least to listen to you, making an argument that this is your, clinical experience space, and that's how you can set meaningful specifications.
Yes, and Liming, I would like to comment on that. I think that's very good question, because we are trying to advance scientific approaches. And something that is important to highlight is that many regulatory agencies have some constraints in terms of the requirements of their guidance documents. They are not guidance documents, they are law. So we also have to overcome the legislations that they have in their countries, and the implementation worldwide of these approaches might be lagging, based on the acceptability and the review of their guidance and legislations. So I think it's important to highlight that. So it's a good start. I think we have noticed that the FDA have favorable acceptability in terms of these approaches, and I think the European agency as well, and Canada. But we have a very good start, and I think we should continue with these approaches, because eventually it will be acceptable worldwide.
Right. Yes, I definitely see the benefit of for a global acceptance of for the approaches, because it will definitely provide the flexibility for post-approval changes. Yes. So it's just we do some investment upfront, but we will gain the flexibility down the road. Yeah. All right. Great. So another question is also about the safe space. Is the safe space approach applicable for all BCS classes, or do the agency may challenge the same for post-approval changes for BCS class 2 or 4?
So, I mean, the safe space approach can be applied to any BCS class, right? Again, ICHM 9 gives you some good guidance with respect to, dissolution profiles need to meet certain criteria as far as, is it a very rapid or rapidly dissolving drug? But even for BCS class 1 compounds where these criteria are not met, you may have to look into establishing clinically relevance if that becomes a liability from a life cycle management perspective. For poorly soluble drugs, as I was trying to explain earlier, the actual ability of the method to really pinpoint whether or not changes in dissolution profiles are indicative of changes in quality, that is very critical. And it doesn't really matter whether you have a BCS class 2 or 4 compounds. The approaches are essentially the same.
Yeah, excellent. So, we have similar questions coming in with regard to the guideline, the reference. Where can they find the reference for establishing safe space? Is there any guideline that has described the safe space?
There isn't truly a guidance out there. I mean, if you go back to the FDA's dissolution guidance from 1996, if you really reread this guidance again, it does stipulate some of the aspects that we have, mentioned today. But unfortunately, this guidance hasn't really then got enough traction, and pushing this forwards into also SUPAC and other guidances that still require you to perform dissolution testing. I think, with some modifications of existing guidance, that could be very quickly fixed. So yeah, long answer short is there is no guidance currently addressing safe space specifically.
We lost Liming.
Oh, my apologies. Yes,
Okay.
I mute myself. Yeah. Okay. So I think that Another question is about what is your experience with the dissolution profiles not reaching 85% in certain conditions? Is F2 still the way to prove equivalency?
So again, it all depends. If you have a dissolution profile, then again, reading through some of the guidance documents that stipulate, well, once you reach a plateau, you stop collecting time points, right? Whether that plateau is reached at 50% label claim, or once you get lower than that, let's say you go below 10%, using F2 doesn't make any sense anymore. So in that case, you may say, I mean, the profiles in this medium just don't show you any release. A lot of times you see actually pretty high variability at these pHs where you see very little amount of drug released. So, I mean, I think it's discretionary. You should make a good judgment call, but again, there's always the there's always the chance that a regulator asks, well, still run F2. So that's unfortunate, yeah. But that's why I really hope we can get away from this multimedia dissolution testing, which in my mind is just, as ambiguous as having an ambiguous QC method.
Yes, so Liming, very quickly, just to say that I know that several regulatory agencies will require the dissolution profile, even if what you are obtaining is like 10%, and basically you are showing solubility instead of dissolution. They still wanted to ensure that due diligence has been done, and that not necessarily we went directly to conduct a BE study exposing the subjects to unnecessary study. So that's the reason that they will be requesting to see the data that was generated, even though it does not pass F2.
Okay, great. Yeah, so I do know that we are at the time 12:30. So, but we still have a lot of questions. We haven't, get to it. And I also have a question for Connie. So perhaps we could close out the Q&A with a question to Connie, and then we can answer the rest of the questions that we haven't, we're not able to get to. Post the response on the IQ website. So the question for Connie is, what is the current, state in terms of implementation of established conditions in the industry?
Yeah, so that's a really good question. ICH Q12 reached step 4 last year, and we're waiting for it to get to step 5 for full implementation. In the meantime, the FDA did run a pilot, and I think there was 10 different companies that participated in that and proposed different established conditions. So there's really good experience in trying out the new tools that are available. We were a participant in that pilot. The product that we were filing did not have a safe space, so we didn't try any of the things that we talked about today, but we do have another product coming up for which we would like to try that once the full implementation takes place.
Great. Thank you so much. Thank you, everybody. Thank you, all the presenters. This is very informative and great presentation, as well as the Q&A session. We have certainly this topic generated a lot of interest. So we will get it back to you. The question we are not able to get to today, we will compile the answers and post them on the IQ website. And if indeed you have any question you would like to, speak to our presenters, feel free to forward your question to Mahalia. With that, I want to thank you. Thank you to the audience, and we will see you, another time.
That's good.
Thank you very much.
Thank you.
Have a great weekend, everyone. Thanks for joining us.
Yes,
thank you.